Hi, my name's John Marshall from McMaster University. I'm one of the Care Gastroenterology faculty. I'm here with Dr. Brian Fagan, Professor of Medicine at University of Western Ontario in London. And we're here to discuss some interesting news that was presented at the United European Gastroenterology Week in Vienna recently. As we all know, biologic therapy has really revolutionized our management of inflammatory bowel disease and improved patient outcomes drastically. Of course, many of the biologic therapies that we use have come off patent, and with this has arisen the competitive molecules, which we call subsequent entry biologics or biosimilars. And arrival of these biosimilars has a number of potential implications for our own practices, but also at a societal level for how we manage our pharmacy budgets and how we deliver healthcare in Ontario and in Canada. Now, the Norse trial was a randomized open-label trial studied in a cohort of patients who are in stable remission on the innovator INFLIXIMAB, which is REMICADE. And they were randomized to either continue on that therapy or switch to a subsequent entry biologic version of infliximab, which is INFLECTRA in Canada. Patients were followed for outcomes. Now, the patient population entered in that trial was a variety of diagnoses, a mixture of rheumatologic conditions, as well as inflammatory bowel disease, and also psoriasis, all followed for different endpoints over the course of the trial. So I'd like to discuss the trial itself and its results, its design, and more importantly, how it might be interpreted and what its implications are for practice here in Canada. Now, as you think about this trial, perhaps I'll ask you, Brian, why do we need this kind of trial now?
Well, I think that, as you indicated, these drugs are similar, they're not identical. And the reason they're being introduced in medical care is really a cost savings issue. And while we appreciate the need for cost effectiveness, we should start with effectiveness. And if two things aren't identical, well then you have to determine whether there are clinical efficacy differences, and the best way of doing that is a randomized trial. So from first principles.
So we'll talk a little bit about the trial, but maybe first of all, what are the strengths of the trial and how it was designed?
Well, it was randomized, and it was designed as a non-inferiority study, and that is both a strength and a weakness of the study. Non-inferiority studies are a little bit odd. Usually in clinical medicine, we wanted to determine whether drug or strategy A is better than drug B or strategy B, and we decide what a clinically important difference is, and we power the study to discern that difference with reasonable confidence. And in a non-inferiority study, it's all topsy-turvy. You're really trying to prove that two things are the same. And in statistics, there's no such thing as identical because of random distributions and statistical testing. So you have to compromise, and you have to decide not what's clinically important, but what's clinically meaningless. And therein lies a first problem with a non-inferiority study is where do you set that bar? So in this case, the study set the bar that we would be willing to conclude that the treatments were identical or the same, that there was no difference between them. If there was a 50% difference in favor of the, no less than a 15% difference in favor of the biosimilar. So you can argue about that. It's really one of the key things about the trial is that many of us would say that 15% in relapse rates across 6 different diseases is clinically important. So if it's clinically important, for a non-inferiority study, that difference is pretty large. And a lot of us would have been happier if we'd said, say, a 7.5% difference, or because that's half of a clinically meaningful difference.
Okay, so maybe we can move a little bit towards the results of the trial, and do you think the results are valid?
Well, the study met its predefined endpoint in that you can put a p-value in a non-inferiority study, and the p-value was statistically significant to say that based on the criteria of a 15% difference with 95% confidence that the results would fall 95% of the time as being less than 15% different in favor of the biosimilar, then it met its primary endpoint. But it comes back to that issue that we started off as an appropriate non-inferiority margin, and a lot of us feel that it was an appropriate non-inferiority margin. So you might ask, well, why would you design a trial that was underpowered? And this trial was supported by the Norwegian government, and it was really, the sample size, from my understanding, was really empiric. They said, we have this much money to do a study, you do the best study you can, and so the sample size of 500 people based on a 15% margin was chosen in that way. And that's probably not the way you want to design a trial for first principles.
Based on convenience and
yeah, and really Norway's a small country, and so you would ask, well, why would you choose 6 different diseases because of all the obvious problems of measuring different things in different ways? That is an intrinsic weakness of the study. But Norway's a small country, so to get the 500 patients, they needed to use all 6 diseases. And therein, I think one of the really controversial results of the study is that when you do subset analysis of the different diseases, actually the point estimate for the Crohn's contingent of patients was over 100 patients, was 12.4%, I believe. Actually in favor of the innovator product, and that's going to be an extremely controversial result because gastroenterologists are fairly narrow-minded and they don't care very much about rheumatoid arthritis, and so but here are the Crohn's results, and this is a 12.5% difference in favor of the innovator. I mean, that must mean the biosimilar is inferior.
Well, I mean, this kind of addresses the extrapolation concern that Health Canada was initially reluctant to
concerned about
about
based on in vitro data of. I neglected to say at the beginning, really the issue around biosimilars is that they are different in their carbohydrate composition, and specifically the fucosylation pattern in this molecule, the biosimilar molecule, is quite different from the innovator molecule, and there are some functional studies that showed that it was behaving different in terms of antibody-dependent cytotoxicity, which is thought to be an important mechanism. So yeah, it is interesting that the results, you could argue, recapitulate the Health Canada's potential objections. But
in truth, we have very few other clinical data in inflammatory bowel disease.
And my real feeling about this is that it, like most subgroup analysis, it's probably a red herring, and that notwithstanding the problems with the power of the study, that the result is probably valid.
Yeah. So, I mean, perhaps we should think, I mean, we've touched on this a little bit, but if you were to design a trial to address this question from first to principles, how would you conduct it?
It's a bit interesting that the regulatory authorities, their take on all of this is I think they're a little bit confused about biosimilars, and they're not following their own guidances. So there's a couple principles that they have implemented with regard to how we go about regulatory approval of this. And the 2 concepts, one is they refer to as assay sensitivity. So that sounds a lot like generic drugs and PK equivalency studies. But what they mean by that is you should pick the disease which has the greatest sensitivity of showing a difference if one should exist. And that would not be rheumatoid arthritis with an ACR20 score. That's a very non-specific outcome measure. It's highly subjective, and maybe I'm biased as a gastroenterologist, but ulcerative colitis has been a real challenge with regard to the pharmacokinetics, and if there were differences in the molecular structure that led to PK differences, you'd see it in ulcerative colitis, and we have a very objective measure in ulcerative colitis. It's called endoscopy, and so I think ideally you would have designed this as a proper non-inferiority study. That's the other thing the regulatory authorities are fascinated by. They'd like to see equivalency studies, which is a 2-sided study. They want to know whether it's not only no worse than, but no better than, which as a clinician I find the latter not really a relevant concept. So, what would this look like if it was designed, if you had 50 million kroner as opposed to 5 million kroner? It probably would involve around 1,300 patients. You would pick ulcerative colitis as the assay-sensitive indication, and you'd use endoscopy as an objective measure, centrally read endoscopy.
Not an easy trial to recruit or to execute.
Life isn't easy here.
That would be the ideal. So, the trial is completed. We have the results, and now here we are in a Canadian landscape. I mean, can we interpret these results in Canada? Can we apply them?
Well,
Is this direct
John,
policy?
the ship has kind of sailed in Canada anyways. I mean, the federal government has approved the drug through the regulatory process using their criteria, and I don't think we can argue with that process. That's what they get paid to do. That's what they're expert in, and they've actually approved the medication. So I think that that point is established. Now, what the federal government didn't opine on was the really key question regarding interchangeability. And the language here matters. So Norse was a switch study, but it was a one-way switch. The patients were randomized either to continue INFLIXIMAB or the biosimilar INFLIXIMAB were randomized. That was randomized, but it was a one-way switch. They didn't switch back and forth multiple times. And in the current environment, that's not an issue that you would be switching between multiple products. It's just two products. But a concern exists that multiple switching, non-medical switching, based on price, would ultimately be a stress test on the immune system. And it's theoretically a way of sensitizing a patient using interrupted therapy because the molecules are different. So the real key here, and what that's referred to, is interchangeability, not substitution or switching. And interchangeability, the federal government actually passed that decision on, both in Canada and the United States, to the province and states respectively. So that is the issue, and it's the issue I think that I'm most concerned about. I mean, the federal government has ruled that they can extrapolate from rheumatoid arthritis to Crohn's disease, and they've done that. So new starts, you will see biosimilars being used in Canada for sure because of the cost issue. But what I'm concerned about is taking stable patients and interchanging the drugs based on cost, where the contract comes up every 2 years and you've got 4 biosimilars, they're bidding on the contract, and you could start out one drug and end up on 2 or 3 different drugs. And I think we need really good data to suggest that that's safe with regard to immunogenicity.
Well, perhaps that leads to my next question. I mean, as you say, the ship perhaps has sailed, or is certainly sailing in Canada. This is going to be the reality of our marketplace going forward. What can we do in Canada to learn from this, or perhaps to...
Well, I think we have to be strong advocates for our patients. And if I'm concerned about this issue, it's a theoretical issue. We don't have any data. We need data. And until I have data, I'm going to not accept interchangeability. If it's forced on me, that's a different issue. And patients and physicians do have recourse. This isn't the Czech Republic, at last time I looked, and where they mandated interchangeability across the board. And so I think we have to take a scientific approach. This is a billion-dollar industry. Surely we can afford a couple $1 million to do a proper study on interchangeability.
So we can study this in Canada. I mean, is there a role for post-marketing studies in Canada?
Yeah, I think so. I think that, and I think the provincial governments and the federal government should hold the feet to the fire of the biosimilar companies and tell them they have to do these studies if they want interchangeability.
And, you know, obviously this doesn't affect just gastroenterology, but our colleagues in rheumatology, dermatology, and perhaps other other areas as well. Do you think other specialists have similar reactions to yours and ours?
Well, John, you will remember we've come from a troubled background with regard to biologic therapy. We weren't particularly clever when we started up. We used these drugs as monotherapy, we used them intermittently, and we sensitized a lot of people. So we've learned the hard way about immunogenicity, whereas the rheumatologists used continuous therapy from the beginning, they used methotrexate background, and they didn't have as big a problem. So I think they're less aware, actually, of this problem than we are.
Well, thanks, Brian. I think we can all learn from your expertise, and I know this is going to be an ongoing area of study, and I'm sure we'll have more to discuss next year. So I think we've learned a lot today about what the landscape is like in Canada and how it's changing with the arrival of subsequent entry biologics. And we've discussed some of the strengths and shortcomings of the Norse trial. Certainly, I think it was a unique opportunity to study the impact of non-medical switching in various disciplines managed with biologic therapies. But of course, it does have its shortcomings. I think Dr. Fagan alluded to the mixture of different diagnoses, to the non-inferiority margin, which was chosen for the study. And of course, all this, I think, raises questions as to how we interpret it. This is a huge area of interest for gastroenterologists across Canada, and the care faculty have been actively engaged in discussing the pros and cons of subsequent entry biologics in Canada, and a number of care programs will arise from this that we hope to bring to you in the near future. Now, in January of 2017, the CARE faculty is sponsoring and organizing a large meeting addressing the subject of subsequent entry biologics. This meeting will be multidisciplinary and involve a number of stakeholders, including patients, providers, and payers. This will be a fascinating discussion across the full spectrum of concerns related to the entry of subsequent entry biologics into the Canadian marketplace. And I would encourage all of you to register and attend this important meeting.